Hey, afternoon, scholars. Good to be back with you here. Uh, I can picture you, and I've got some comments, but I better keep them to myself. And I better get going on this video. This one's going to take a couple hours. No, it won't. Well, hopefully you finished up that first section, the first 4 pages, pretty well with that last video just before this one. We're going to see if we can get through the rest of it today, but probably we'll just get through skeletal muscle, and we'll save a little bit for Monday when you come back after the break, after Good Friday. Carol gave you a couple of assignments in a worksheet, and that worksheet should also help you understand some of the material today a little bit better. I think today it's probably going to be some physiology, so that's function, and function takes a little more thought process than just the structure, knowing the structures do. So let's see how we can do today. So we're going to start on the top of page 5 on your outline with the organization of thin and thick filaments. Thin and thick filaments. So first of all, you have to recall which filaments are the thin filaments and which filaments are the thick filaments. Well, you're correct. The actin would comprise all the thin filaments, and the myosin would comprise the thick filaments. So they're organized in a way that creates this striation look of a skeletal muscle cell. The dark bands that you see in this striation would be called the A bands, and they will contain the entire myosin and an overlapped part of the actin. And I'll show you this as soon as we get done with these 4 points. The light bands are just the thin filaments or the actin filaments. So what makes the dark bands darker is you've got 2 filaments filling up that section of the cell or the fiber. And the light band is just when one myofilament, the actin, is in that area. So let's go back a couple of slides, or I think it's the page prior to this one on your outline. And if you look right here at the top of this figure, you can see this myofilament. Here you can see a dark or an A band, and in this area here is the light or the I band. And you can see in this dark area how there are more myofilaments, actin and myosin, which makes it dark. Here you just see one myofilament, and since this is the I band and there's only one myofilament, and it's which one? The thin one. So this is only the actin. Only the actin. And this is an area that we're going to take a look at in a little more detail in a minute, because this area from this jagged or kind of a little Z-shaped line over to this Z-shaped line or Z-disc, we're going to see later this is called the Z-disc. From here to here, that is the main functional unit of a muscle fiber. And we're going to take a look at that a little bit more, going back to where we just left off. So let's go back. We talked about here the organization of these thin and thick filaments create the dark bands or the A bands, A, dark, and the light bands, I, light. The dark band, again, is where we have the combo, the overlapping myosin and actin. And there's obviously other things in here as well, but this is going to be the basic aspect that you'll need to know for 211. All right, now we're going to take a look at the functional unit of that muscle fiber. And I just showed it to you when I said it was from that Z disc or that jagged area between the actins over to the jagged area between the other actins. And this unit, this structural unit, functional unit of the skeletal muscle, it's both structural and functional in this case, is the sarcomere. Remember what that prefix sarco means? Sarcomere. This is the muscle cell functional unit. As I said, it's between these two Z disks, and just below this on your slide you'll see this picture, and we'll take a look at this in a little bit more detail in just a second. But here's that Z disk, and again later please compare this one to that one we looked at just a couple minutes ago that's on the previous page of your outline. So it's not as detailed, but you can get the general component parts, and it's good to look at these in a couple different ways. So, here we go right here. So the sarcomere is the area between the two Z disks. And remember that these Z disks have in it the dark and light bands, and that those dark and light bands repeat themselves all through that myofibril. So there's many myofilaments. So there's very many sarcomeres within a myofibril. So here you see the myofibrils contain multiple and repeating end after end after end sarcomeres. Now when the sarcomere shortens, here's the critical part, when the sarcomere shortens from the Z disks being pulled together, when the Z disks are pulled together, that's when the muscle contracts. So those cross bridges are formed from the tail and head of the myosin, and they will attach to the actin, and they will pull the actin together that they're attaching to. We'll show you this in a little more detail here. Again, here's the sarcomere from a Z disc to a Z disc. Here's that Mittelscheibe, the middle in German line, and this is in the middle of this A band here. The A band is where there's an overlap of the actin and the myosin, actin and myosin, actin and myosin. And remember, there's many of these within a muscle fiber. And then over here is that I band. And if you look above the I band, it's only actin, 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 actin. This here is a little protein that also helps with the contraction. It's called titin. You do not have to know that for this class today. But it's another protein, just like actin and myosin. It plays an important role, but not the big deal role like actin and myosin. So we'll be looking at this, and this should look a lot like what Carol gave you in class today. All right, now we're going to take a look in a little more detail of the skeletal muscle contraction. So when muscle fibers shorten, there's an interaction between these thin and thick filaments. They get pulled closer together is what's going to happen. And so the mechanism for this is called the sliding filament theory. Filament. So it's really the sliding myofilament theory. The myofilaments, the actins and the myosin, slide, and that's what causes this contraction. It's kind of like the electric slide. Many of you know that. They were thinking of calling it the sliding filament, but cowboy songs don't work out with sliding filament theory. So anyway, again, during the contraction, the thick and thin filaments interact and slide past each other. This causes the following changes within each sarcomere, and you're going to have to know these parts too. So let's go back to look at this area here. When these actins, see, these heads will attach to this actin, these heads will attach to this actin will come in here, this actin will come in here, and then what will happen? What will happen here? Well, let's take a look. As these actins come in to this area, we will see that they will come closer to this M line, and that this A band will all become dark because of them sliding over. So the width remains the same of that A band, but it fills in. The H zone is this area where the actin wasn't at the beginning, but is now filled in. This H zone will actually disappear as these get pulled in. So H refers to heller, which means a bright zone in German. So while a lot of the terms we've talked about in the past have been Greek, a lot of the theory on muscle contraction was discovered by scientists over in Germany. Hence, we'll get these different letters that appear on the muscle contraction. The Z discs, the Zwischenschreib, which means between, the Z discs are between the actins, and each sarcomere will move closer together. And the sarcomere narrows in length, and the I bands also narrow. The length of the thick and thin filaments never change, however, when the muscle is contracted or relaxed. And if you go to, it's really page 298, you can see more about the sliding filament theory there, more figures that show what we just discussed. So kind of going back here again, the actin never changes in length, the myosin never changes in length. The A band never changes in length. The I band will get smaller because during that contraction, the Z discs move closer together. The Z disc is attached to this actin. This actin is ratcheted towards the middle. Hence, this whole Z disc will come towards the middle. And the same thing will happen on this other side. So take a look at those constants. Take a look at those 5 areas we just talked about here. What happens during the contraction? Study that area. Study this material here. Okay. Next we have another term we have to define, because in order for any of that contraction to take place, we need a stimulus. Most of the time the stimulus is from a nerve. In fact, it's called a motor nerve, because this nerve, we have 2 types of nerves. One are for sensory, one is for motor. The motor nerve causes movement. The nerve's function is to fire a muscle to cause movement. So we'll have this neuromuscular junction where the nerve comes close to the sarcolemma of the muscle cell. So the muscle contraction begins when this motor neuron sends an impulse to the muscle fiber. So as I said, this neuromuscular junction, let's see where I want to show you this. Let's see, neuromuscular junction, maybe this one, nope. Sorry. Here it is. Here's a motor neuron. This is on your, off to the right of this section on your outline. Here's a motor neuron sending axons. You don't need to know that. Just know this is a motor neuron. And off the end of the motor neuron, there are a number of ends of the nerves that will be stimulating different muscle fibers. So here's 3 muscle fibers being stimulated by one motor neuron. Each area where that motor neuron comes close to the sarcolemma, or the outer part of the cell, is going to be called the synaptic cleft, or it can be, there's an area in there called the synaptic cleft, but it will be called the neuromuscular junction, where it comes close there. All right, let's see, which way do I go? I need to go this way now. Oops. So the neuromuscular junction is the region where the motor neuron comes in close proximity to the muscle fiber on that sarcolemma. Here's a closer look at this connection, and we're not going to be so concerned with this detail. Here's kind of a neat picture, though, of the endomysium that covers the sarcolemma. You know, the different coverings, concentric coverings of the muscle we talked about earlier, the endomysium, the perimysium around the fascicle, and then the epimysium around the entire area there. So I'm going to go down one more here. So how does a muscle contraction happen? Well, that motor nerve sends an impulse and it causes ACH, acetylcholine, to be released into the synaptic cleft. So that you can see that close up here. So this sends, there's a nerve electrical impulse comes down, hits the endomysium and the sarcolemma, and that causes ACH to be released from this area into the into the sarcolemma, into the sarcoplasm. So the acetylcholine is an important starter for this muscle contraction. And this is what happens. And this is just part of what happens. In exercise physiology, they'll certainly go into more detail. So, and some of this should be a little bit repeated from what we talked about Monday, I think, on the video. So, calcium is released from the terminal cisternae. So that impulse hits the sarcolemma. They're spread around the sarcolemma via the T-tubules. Yep, the T-tubules. And the T-tubules will stimulate the calcium to be released from the terminal cisternae. And when the calcium is released, it will actually go to an actin. And on the actin, there is a receptor site for the head of the myosin. This calcium causes that receptor site on the actin to be open, to be receptive for the head of the myosin to attach there. So we need acetylcholine to come in. This triggers the release of the calcium. When the calcium is released, seeks out that actin and that receptor site, frees it for the myosin to come in and form the cross bridge. Here we can see that a little bit here too, where the acetylcholine's released, allows an impulse to be triggered down the T-tubules. The T-tubules cause the calcium to be released from the terminal cisternae, and then that calcium will attach on the actin to a receptor site, which opens that receptor site so that the head of the myosin can attach there. Those are the key aspects so far. Then, in the presence of ATP, made in the mitochondria in the sarcoplasm, ATP, which is the energy needed, the myosin can then cycle through the attachment. It's going to attach its heads to the actin. Technically, it pivots and pulls the actin in, causing the contraction or shortening of that sarcomere, in which happens along the entire muscle fiber. So that entire muscle fiber then shortens, and then a number of these muscle fibers do that same thing, and so the muscle shortens. So I hope that kind of makes sense. Then when the ATP is no longer there, it causes a detachment of the head of the myosin from that actin. There's muscle relaxation, and the stage is then automatically reset for another muscle contraction. It's important to note that ATP and calcium are also both necessary for the relaxation of these muscle fibers as well. So I hope that kind of makes sense. got an assignment for you for Monday that should also help you understand this a little bit better. All right, now let's take a look at a motor unit, because if one muscle fiber, or even if 5 muscle fibers can contract and shorten, I don't think that's going to cause that whole muscle to shorten. So motor units are going to be the single motor neuron like we saw, and all the muscle fibers that it controls. All the muscle fibers that it controls. In the example that we saw earlier, there was 3 fibers that are controlled by this one motor neuron. Well, sometimes there can be up to 100 muscle fibers controlled by one motor neuron. So more efficient, but what we'll find generally is that the more motor nerves, the more there, I got that in there. The more muscle fibers that are controlled by one motor neuron, the less fine, smooth motion you're going to get. So if you just contract your knee, it's probably a motor neuron with a lot of muscle fibers, because that's not a very fine movement. But if you're playing the piano, moving your fingers at the speed of light, then there might only be 3 or 4 or 5, certainly less muscle fibers that would be innervated by that one motor neuron. So a motor unit is going to be that motor neuron and however many muscle fibers that it will stimulate. So that will vary. So let's see, that's what we were saying right here. So fine movements have less fibers being controlled by one motor neuron. Gross movements have more fibers being controlled. And it makes sense too. Larger muscles will have more motor units. Smaller muscles will have less. What each muscle fiber obeys, though, no matter what the size of the motor unit, is that there's this thing called the all or none principle. A muscle fiber will completely contract or not at all. And what that means is, throughout the myofilaments of that muscle fiber, all the myofilaments will contract or none will contract. It's called the all or none principle. Now, one thing that you need to remember is that within the myofilament, all those actins and myosins will contract. But in all the myofibrils, not all the myofibrils will have to contract to cause the whole muscle fiber to contract. It may take 80% of the myofibrils within a certain muscle fiber to contract, to cause contraction. So there's always some that will not fire because they will need to have to be ready for the next firing. So when a motor unit is stimulated, all the muscle fibers under its control will act or fire. It's called the all or none principle. It won't just go part way. When you move your muscle part way, you're controlling the movement, and only so many motor units have to fire to get a partial contraction. If you're doing a maximal contraction, you will recruit more motor units and more muscle fibers. Hope that makes sense also. Okay, types of muscle actions. And I'm going to have Carol do most of this work because when we see this shortening of a muscle occur, when that sarcomere shortens, that's a contraction. But we know that in some muscle actions, that the muscle actually lengthens while it's being electrically stimulated. How can it lengthen in that even these cross bridges form, the actin is pulled across the myosin, and so the sarcomeres shorten. How can we have what's called an eccentric action, where the muscle actually lengthens during an activity, or during the firing of a muscle? Well, that's just being looked at now, or it's been looked at for a while, but either the microscopes or they haven't been able to determine what's really going on. So what they're thinking is happening, this is what I've read most recently, is that the actual tails of the actin, even though they're ratcheting, they continue to ratchet the actin, the tails are being pulled and lengthened. So the tails are getting longer while they're attempting to actually pull that actin in. So in a normal action where we see the two Z discs coming together, these Z discs will not come together in an eccentric action because the tails of the myosin are actually being stretched based on the action that's trying to be performed. And Carol will also demonstrate this isometric here. So you can maybe pause this now for a minute and try to do that. Okay, and when you come back, I'm going to be talking about types of skeletal muscle. So let's see, types of, I'm just going to take a look at my notes here. So it depends on where we are on Wednesday, if you have time to go into types of skeletal muscle. Otherwise, I don't want to throw this out there, but I will. This can wait until Monday. So I'm going to let Mrs. Moss be the decision maker there. No, not you. Who is that? Who's that over there? Kurt? No, you're not going to make the decision today. Thank you. Okay, so here's skeletal muscle. We've talked about how that works. Well, now there's actually 3 types of skeletal muscle that you have to know. Earlier we said there are 3 types of muscle: cardiac, smooth, and skeletal. Well, now I'm going to tell you that there's 3 types of skeletal muscle. And based on the types of muscle fibers. So skeletal muscles will be comprised based on the types of muscle fibers that you have. So this is often done with something called a muscle biopsy. They can go in with a needle, pull out some muscle cells, and they can look at the muscle fiber types. And we'll talk about these 3 types. And how these 3 types are actually mixed together will determine the speed of the muscle's contraction and how long that contraction can be sustained. So these fiber types are all going to be relative to some sort of speed of contraction. So let's see what they are. Oops. Here they are. One type, type 1, type Roman numeral 1 is what they're called, is a slow fiber, sometimes called slow twitch fiber. It's a slow twitch muscle fiber cell. Note that it's slow and it's oxidative, meaning that it needs more oxygen. It's going to need more oxygen. There's another fiber type that's called the intermediate, and it's going to be called type 2A. Type 2A. It's fast and it's aerobic. So aerobic means that it needs some oxygen, but not as much oxygen as the slow muscle fiber type. Lastly, we have fast twitch fibers. Fast twitch fibers are also called type 2B, and they are fast and they are anaerobic. If we put AN in front of a term, that means without, so they actually work without oxygen. They work so fast they don't need any oxygen to sustain that contraction. Now notice here, type 1, and then these are both type 2. Well, I'll tell you, back in the dark ages when I studied this material as a young scholar, we only had 2 muscle types. They were called slow and fast, type 1 or type 2, oxidative or anaerobic. So somewhere along the line, some other scientists, researchers, determined that, hey, there's some of these other muscle fiber types that we're seeing here. They require a little bit of oxygen. They work a little bit faster than these slows, but they're not quite as fast as our fasts. So either these things evolved in the last 40 years, which I doubt, but they were discovered with better research methods. So we have 3 fiber types. And here are some characteristics of some of these. You'll need to know some of these characteristic types. So the fiber characteristics: slow, intermediate, fast. Need to know the type 1 and oxidative, type 2B, type 2A, anaerobic and aerobic. Need to know all that. Now let's go down and take a look, and you will also need to know let's see here. Let's go down to the color of the fibers. Yes, colors are always good. And this is what they actually look like under a microscope. The slow fibers look redder. Well, why do they look redder? They're more oxidative. We'll find out later that the oxygen is in the hemoglobin of red blood cells. So there are more red blood cells that will be working with these fibers. Hence, they're red. The intermediate fibers are also characterized here as red, but we'll take a look in a minute. They're actually going to look more gray, maybe a pinkish, but not a dark red. And then lastly, the fast ones do not need any oxygen, hence they're white or pale as a looking cell. So you can see these cells by what they look like, their color. Let's take a look at their fiber distribution. Where are they found? Well, they are found in the greatest abundance, this is the slow, in the postural muscles of the trunk. So the low back muscles, the abdominal muscles are slow twitch fibers. Even the calf muscles in the leg. The calf muscles in the leg are actually more slow fiber in nature. Because as you lean forward, your calf and gastroc get put on stretch. So that would initiate these to fire a little bit. Also, let's see here. The intermediate types, they're found in the greatest abundance in the lower limbs. So most of the leg muscles are intermediate. And then lastly, the fast fiber types are found generally in the upper extremity. And again, remember, this is a ratio of fibers. So, for example, a biceps might be 60% fast, 30% intermediate, and 10% slow. So it's a ratio of so many. We do know that in eyes, eye blinking, pretty fast activity, it has no slow fiber types, the muscles that cause eye blinking. But the muscles in the lower limbs might be 40% intermediate, 30% fast, and 30% slow. Some of them might be 50%. And it's going to vary from individual to individual what their fiber types are. It's known to be genetic by nature, and now we're finding that perhaps this intermediate fiber can take on characteristics based on what's needed. It can take on the characteristics of a slow fiber, or maybe of a fast fiber as well. So which do you think would contract fastest? Here's another area. This is for sure going to be the slowest. This would be fast, but not as fast as the fast types. And then the last thing we want to look at is the primary fiber function. Well, if I have slow fiber types, you're going to see that mainly in the marathon runners, the distance runners, distance swimmers, maybe most swimmers, because even though the sprinters in swimming move pretty darn fast, the amount of times that the muscles contract isn't the same when you compare it to, say, a sprinter in running. A medium duration is the fiber function of this intermediate type. Moderate movement, kind of makes sense. And then fast fibers would be the sprinters or lifting weights, something that you do very ballistically. So those characteristics you should know from this table. That would be the fiber characteristics, the color of the fibers, the contraction velocity, the fiber distribution, and the fiber function. Next, here's a picture, here's a slide of this is a muscle biopsy of a cell, or of a muscle fiber. And in these muscle fibers, or of a muscle, sorry, this is a slide of a muscle. And it's got different types of fiber types in it. Slow fibers are these darker red ones, so every dark red one would be a slow fiber type. Intermediate, I, is more of the grayer ones, and you can see there's a lot of gray in here. And then the fast fibers would be the white ones. White, white, white. So what you could actually do is count up the dark red, count up the kind of pale colored ones, and count up the white ones. And based on that ratio, you would know if this is probably a slow, an intermediate, or a fast fiber type. I'm guessing, looking at it, that there seems to be more of this kind of gray colored, pale colored, gray or pink colored cells, so it would make it intermediate. That's just my guess in looking at the types of fibers in this muscle. These are things we've already mentioned, how they're comprised of the three types, where they're distributed is on that chart we just saw. Let's see here. Let's continue and take a look at how exercise affects skeletal muscle or lack of exercise. Well, muscle is a tissue that can get smaller. Reduction of muscle size and power is when we have muscle atrophy. Trophy means growth, without growth. And that will occur as actins and myosins may go away if they're not used. While you'll still have the same amount of muscle fibers, the size of the muscle fiber will change because the actins and myosins that are no longer needed will not be produced by the body. So that's muscle atrophy. Muscle hypertrophy is an increase in muscle fiber size. And it turns out some recent research shows that actually there may be an increase in number of fibers as well. So the more we learn about doing research on different things, the more we learn about that thing. In this case, muscle fibers. We seem now to be having seen an increase in some subjects during some research. I don't know the research, so I can't tell you more about it. But I know for the longest time we were taught to teach that hypertrophy is not due to an increase in muscle fibers, but an increase in the actins and myosins within a muscle fiber. Now, if we get an increase in muscle fiber, if we get muscle hypertrophy, we'll also need to have more mitochondria, which will increase the number or the ability to produce ATP, which we need for a contraction. The myofibrils and myofilaments increase in number, resulting in that muscle size, and that's what I said at the beginning. All right. Another muscle type that we talked about earlier was cardiac muscle fibers. They are also striated, like the skeletal muscle, whereas the skeletal muscle was considered to be multinucleated. In cardiac muscle, there's only one or two nuclei. I don't have a picture of the Y-shaped branches on these cells, but if you take a look on page 311 in your book, you'll see how these adjacent cardiac cells attach in this Y formation to form junctions, and these are called gap junctions. and how they are adjacent. They become adjacent through this term of intercalation, or the cells are said to be intercalated. Cardiac cells are. And what this allows them to do is to have an automatic rhythm, so that when the heart beats, it generally beats in this automatic rhythm. It's because one muscle cell, one cardiac muscle cell, innervates the next cardiac muscle cell, innervates the next cardiac muscle cell. So you have this sequence of muscle cells innervating the adjacent cell. This is very unique. Smooth muscle and skeletal muscle certainly don't work this way. So that's why they're able to generate muscle impulse without nervous stimulation. So for example, if you can volitionally control your motor unit, so you can tell your biceps to stop when it's contracting. Okay, now I want you to try to do the same thing with your heart. Tell your heart to stop beating. It's not going to happen because it's not controlled by you. It's an involuntary control with this autorhythmic response. Smooth muscle are found in the walls of viscera and blood vessels. They're short fusiform cells. So what's a fusiform cell? So a fusiform cell has tapered ends, wide in the middle. So it'd be tapered at an end, wide in the middle. Now this is actually a muscle, but that's what the cell looks like for a smooth muscle cell. They only have one centrally located nucleus, and they have no striations. No striations. There are thin filaments attached to dense bodies. So that fusiform look is a dense body in the middle with thin filaments on either end. And these also work involuntarily. They work involuntarily. And that's what I got on muscles. The end. Fire up. Have a fantastic weekend. An incredible, great Good Friday. And Easter as well. All right. See you next week. Or maybe not. No, I think Carol's got you. I'll be talking to you, though. Take good care. Bye.